Welcome back to the Genetics Lecture Series. We took 1 week off, but now we're back to business. Once again, I am Drew Wico, and today we're going to take a look at how genetics is portrayed in pop culture. If you have any questions about what we cover today, please leave a comment below. We are going to first revisit the material on building a pedigree, then we will jump into pop culture genetics. Lecture 6 expanded on how we build our pedigrees, primarily focusing upon the rules that must be followed. It seems like an easy process, but violating rules of pedigrees can create a lot of confusion, so be sure to stick to them. Remember to make the correct shapes to note who is a male and who is a female, and that it's important where you connect all the shapes. Siblings are connected from above the shape, parents are on a different plane or level than their children, and mating pairs are connected by a direct horizontal line. A major rule that is typically violated is labeling the proband, who we use an arrow for. This is important because if we want to show the pedigree to a healthcare or genetics professional, it helps them know where you are, or where the person of interest is in the pedigree. It serves as a coordinate system or a launching point for them. As for common mistakes, we talked about the largest one, which is how cousins are discussed. First, second, third cousins and how many times they are removed gets really confusing. But we talked about how each of these scenarios would work. If your mother has a sister, which you would call your aunt, the aunt's children would be your first cousins. If that cousin has a child, they are your first cousin once removed. If you have a child, your first cousin once removed would be your child's second cousin. It all has to do with how it connects back to the common ancestor. We also covered how siblings and mating pairs sometimes get misrepresented on pedigrees, so be sure to revisit those rules. If you're still not comfortable with all the rules, don't be afraid to revisit Lecture 6. I myself even have to go back to my own notes on pedigrees from time to time, so there's absolutely nothing wrong if you don't have a total grasp on it yet. We will start today's lecture on genetics and pop culture with the film that comes up most frequently in conversation for me when I mention to people that I study genetics. The 1993 film Jurassic Park brought plenty of ideas to the foreground of what might be possible, especially when considering how much genetics was beginning to advance at the time. Jurassic Park was all about bringing the dinosaurs back as an attraction. How did they explain this within the film? The concept was that pesky mosquitoes went around and sucked the blood out of the dinosaurs, got fossilized in amber, freezing them in time. The scientist who discovered these fossilized mosquitoes would extract the blood out of these mosquitoes, sequence the genome in the DNA, and fill in the gaps with modern-day relatives of dinosaurs. They would then cross the genome into a frog, and then the frogs could lay dinosaur eggs, and thus the dinosaurs were brought back. Magic, huh? I will say, regardless of what negative I might say about this film's approach to science, it was a really exciting movie, and I love it still to this day. It was also really educational with Mr. DNA on the right of our slide right here, who attempted to do kind of what I'm doing right now, explaining the confusing science in the simplest way possible. But let's take a step back and start to think about if bringing the dinosaurs back like this is even likely, let alone possible. I hate to burst everybody's bubble, but this is nothing but science fiction. We've come a very long way with genetics, even from when this movie premiered, but we still can't do this. We can sequence some of the DNA, read the DNA, of the dinosaurs and learn more through what is found in their blood, but there are multiple issues with their science. First, although DNA is incredibly resilient from decay, it still does decay over time. Factors like temperature, moisture, soil chemistry, and other factors help speed up or slow down the rate at which DNA begins to fall apart. But DNA has an approximate half-life of 521 years. This graph on the left side of our slide will help us walk through what this means. This means that in 521 years, half of the DNA has degraded, shown by that green point, the 0.5. In another 521 years, another half of the existing DNA degrades, so we're now left with 1/4 of our original DNA, or 25%, shown by the red line. Based on this math, and our common knowledge that dinosaurs roughly existed around 65 million years ago, and longer back, basically no DNA would be left by this time. You might be asking, but it's fossilized. We see that the mosquito is intact. Doesn't that protect the DNA in the blood? The answer is sadly that when being fossilized in amber, it protects the exterior, but not soft tissues, which includes the blood. We don't get our dinosaur DNA to revive our prehistoric reptilian friends. You can see on the right of this slide that this mosquito, which is actually fossilized in amber, is somewhat transparent, showing that the hard tissue does stay because of the fossilization, but the soft tissues, like the blood on the inside, don't stay. This is before we even get to the complicated issue of crossing dinosaur DNA with a frog's, which we have no evidence of that it would work. Organisms are incredibly unique, and although we share many proteins and parts of our genome with other animals, it doesn't always have the same effect between different animals. This is why so much research is done in rats and rodents, but might not have the same effect when we transition it to human drug research. So, we can't bring back the dinosaurs from extinction. What about if we had a living dinosaur instead? Could we clone it? This leads us to another prominent moment in the history of genetics. We showed that we can't create life from nothing, but can we effectively copy and paste it? On the left, you can see a cute little sheep that gained some notable fame in the late 90s, named Dolly. Some of you might have seen Dolly at one point in your life, but Dolly was the first successful cloning to ever happen in a mammal. Dolly was cloned from a sheep's mammary gland cell, and she was named after Dolly Parton because, well, scientists have a funny sense of humor. I won't provide their exact quote on the reasoning, but it's on the Wikipedia page for Dolly and is an accurate quote from the 1997 BBC article. Dolly was a wild success, but how was her cloning possible? Here is a diagram on how they cloned Dolly. In short, I know it's very confusing, they took the mammary gland cells, allowed them to grow in a culture, or something that allowed them to grow multiple times over, took the DNA out of the growing cells, and transferred it into an egg that had the nucleus removed. One fertilization later in a surrogate, and Dolly developed and was born. Now, this further shows the issue with Jurassic Park because they still used a sheep in Dolly, although it was another species. Frogs are not dinosaurs, so this is yet another argument to why they may have been unable to accomplish what they set out in the film. How Dolly was cloned was a massive feat, but there still arose issues. Dolly had a shorter than normal lifespan and died at around 6 years old, much shorter than the average 12 years of her species, Finn Dorset sheep. Dolly died of severe lung disease and arthritis in 2003, and one of the theories is that the DNA had already been damaged a certain amount before it was implanted into this new egg. This is believed to be one of the reasons that she was more susceptible to illnesses, and how we started to study DNA damage a bit more. But scientists aren't 100% sure that this is the exact reasoning. Overall, Dolly was a wild success and has been talked about immensely. Dolly is still a hallmark in most genetics textbooks. But even with her success, it provided many new details on how daunting the task of cloning truly is. Another popular film covering genetics is GATTACA, a film about a man in a futuristic world who wants to be an astronaut, yet is deemed unfit due to his genetics. This movie shows concepts that have been discussed for years, like genetic engineering. In this specific instance, it involves couples meeting with a geneticist once they find out they're pregnant. Different from reality, where, in this movie, a geneticist helps them choose certain variables about their child, from eye and hair color all the way to athletic ability. This, in turn, brings up concepts of genetic discrimination, where people are seen favorably or unfavorably due to their specific genetic makeup, as well as genetic determinism, where people are effectively locked in to a certain life or fate due to their genetic makeup. Without spoiling the movie, I'll mention that they also cover forensic genetics, which you might have also heard of on the news when talking about DNA at a crime scene, or on detective shows like Law & Order. But taking a step back from what the movie shows, how much of this is really applicable? So some of these concepts may sound like they are totally plausible, while others seem outlandish. So let's dig a little bit deeper. First is genetic engineering and gene selection to make designer babies, as some people have called it. Is this possible? Well, the ability to edit genes is incredibly new, and has almost never been used in humans. The concept of gene editing is one heavily disagreed upon due to the ethical implications, as well as how little we actually know about genetics in humans. Think back to our very first lecture where I mentioned that we have around 20,000 genes, and that they all interact with one another to make very unique outcomes. What happens when one changes just a tiny bit? It can cause a cascade of problems. This idea worries many scientists, since we're still trying to make sense of the human genome day by day. However, there has been one instance of gene editing that we know of, and you might have actually heard of this if you listened to my interview with Aaron Croyle. A scientist in China decided that he wanted to edit the gene for HIV susceptibility. By editing it, he would increase the resistance a pair of twins had to HIV, lowering their chance that they could get it in their lifetime. That's really cool, right? The issue, however, is that the same gene is responsible for immune functioning. We don't know exactly what it does, so the two children might have effects to their immune system throughout their life. To make matters worse, the scientist is no longer keeping up with the children and doing follow-ups, meaning we don't even know what effects this change will have. When this genome editing happened, there was a large uproar from the genetics community around the world for violations of ethics and an overall avoidance of what should be done in research. Moving on to genetic discrimination and determinism, this is sadly something a bit more prevalent and well known, but not exactly in the way that the film portrays. Genetic discrimination has been outlawed in the United States through the passage of GINA, which we've discussed in previous lectures. Eugenics has also previously been an issue not only in world history, but also American history. The sad truth is that many states permitted sterilization of certain groups shortly after the year 1900, such as individuals affected by disabilities or individuals with cognitive impairments. This did change heavily around World War II, when a certain world leader was gaining his own attention for promotion of eugenics and elimination of certain ethnic and social groups, changing the opinion within the public around the United States. It wasn't until 1978 that sterilization was outlawed by the federal government. Since then, we cannot deny that there is still discrimination that exists to this day, but we don't see it to the level of genetic discrimination that is portrayed within this film for simply having a common variant of an allele. Finally, the film covers forensic genetics. This is something commonly discussed in crime shows, but is it actually realistic to be able to determine who did the crime from a single hair at a crime scene? If the follicle of the hair is attached, the part that actually attaches to your scalp, then it is possible. Otherwise, it's not. This is because the follicle would contain DNA since it's still a living cell, while hair is solely made of dead cells where no DNA would be found. But tracing DNA back to an individual is actually a very possible concept. What forensic scientists do is look for certain markers in your DNA where people have multiple copies of a sequence. There are many of these in our genome, so when they see that there are 6 copies in spot A, 9 in spot B, and so on, you can hone in on the match to the DNA. Now, it is totally possible that some people do have identical numbers of these copies, especially if they're related, but the probability of this otherwise is incredibly low. Some forensic agencies also do band matching on PCR, which we can see on the bottom of our slide right here. What they do here is just line up the bars to see if everything matches. So maybe GATTACA wasn't too outlandish. This lecture covered a couple of films that have been popular within the field of genetics, but I want to remind you to take them with a grain of salt. These are movies, so their primary purpose is to entertain you, and to take you to a new world overall, not to replicate science exactly. We can see this through some of their accuracies that they have that go along with their inaccuracies, but they're just trying to tell a story at the end of the day. That's why it's called science fiction. There's a piece of fiction in the story. Next lecture will be our final lecture of the genetics lecture series. We will be revisiting many of the topics we discussed to help you appreciate how far we've come in 8 lectures, building them all off of one another and in a single example. We will look at pedigrees, inheritance, and other variables to see what questions we can have answered. My hope is that by going through it in this way, you can understand how the conversation would go with a genetics professional and prepare you for those discussions. Once again, if you have any questions or comments, please leave them below. Thank you once again for joining me, and I hope you enjoyed this lecture.